everybody, Professor Davis here, and we are going to be looking at the functional anatomy of prokaryotic and eukaryotic cells. And we're going to focus mostly on prokaryotic cells in this particular chapter. But we are going to do a comparison of prokaryotic and eukaryotic first, and then the next video will focus in on eukaryotes. So if we take a look here, there are going to be some distinct differences that we do see in prokaryotic and eukaryotic cells. For one, the groups or organisms that are part of that. So prokaryotes are going to be made up of bacteria, and we're not going to talk about archaea here. Archaea is one of those that doesn't fall into the category of being pathogenic, and so we're not going to discuss them. In the eukaryotes, though, these are going to be the protista, the fungi, plantae, and animalia. So those are going to be those four kingdoms that we talked about in the previous video. If we look at the DNA for prokaryotes is not going to be inside of a nucleus. It's not going to be bound by a membrane. And so because of this, we call it the nucleoid region. Okay. And the DNA is going to be in a single chromosome. This chromosome, though, is circular and it's very large. And so it kind of wads up like you can see here in the picture. With prokaryotic cells, there's not any membrane-bound organelles. They do have ribosomes, but their ribosome structure is different than the ribosomes found in eukaryotes. We say that they have a 70S structure, and this deals with the two subunits that make up a ribosome. For eukaryotes, the DNA is going to be bound inside of a nucleus. So not only do you have the outer membrane of the cell, you also have a membrane of the nucleus, and the DNA is inside of that. These chromosomes are associated a lot of times with proteins called histones, and they are going to wrap around it. And you're going to see that they normally have multiple chromosomes, not just one. We also see that eukaryotes have various membrane-bound organelles. They have things like the Golgi apparatus, the rough ER, the smooth ER, the mitochondria. They have a lot of those little structures that we call organelles that help do different jobs in the cell. They also do contain ribosomes, but their ribosomes are a little larger in their subunits, and we term them as being 80S. And we'll talk a little bit more about those when we get to ribosomes. So this just shows you kind of the difference between prokaryotic and eukaryotic cells. Now, sometimes we do see that they have a cell wall. Now, in prokaryotes, they almost all have a cell wall, and that cell wall is going to contain a chemical called peptidoglycan. Now, there are a few exceptions to this. When we talked about tuberculosis in the previous video, those myclonic type of bacteria, but most of them do have peptidoglycan. Now, in eukaryotes, there may not be an actual cell wall present. In animals, they don't have it. It's absent. But in plants, it's made out of the chemical cellulose. And in fungi, it's made out of chitin or chitin. This is going to distinguish between those groups. Now, what does the cell wall actually do? It's normally protective. It's an extra layer that protects the plasma membrane, which is underneath. All right, how do the cells then divide? We do see that prokaryotes are going to divide by binary fission because they do not have a nucleus. Since they don't have a nucleus, they're going to divide into two, which is the binary, and fission means to split. In eukaryotes, we call this mitosis because the term mitosis means nuclear division. Since they have a nucleus, the nucleus divides in two along with the cell. Now, one thing to note about both of these types of cell division is they are considered asexual. The cell can divide on its own, doesn't need a partner to do this. So if we look at the idea that these are asexually reproducing, they are going to be genetically identical to the parent cell. All right. And so that is part of what makes that difference. Again, we can't say that bacteria is going to be doing mitosis because bacteria does not have a nucleus. So cell organization, when we look at prokaryotes are going to be less complex. They're normally going to be unicellular organisms, and they're smaller in size when compared to eukaryotes for the most part. Eukaryotes, because they contain all those organelles, they are more complex. They're going to be mostly multicellular. Now, again, protista is an exception. And they are about 10 times larger in cell size than what you see with the bacteria or the prokaryotes. So they are larger in structure. So we're going to spend the rest of this video focusing in on the prokaryotes. The next video will then go into specifically the eukaryotes. So with the prokaryotes, we do see that they have the domains archaea and bacteria. Now, archaea does not cause any disease, and so again, we're not going to discuss them much throughout this course. But bacteria is going to be a very, very big focal point in this course. A lot of times with prokaryotes, we are going to ID them based on their cell shape and their arrangement. And this is going to be done by staining them and looking at them underneath microscopes. We do see that there are some basic shapes that are present in the group bacteria. These include round shapes, rod shapes, and helical shapes. And we have some special terms that go with this. So if they are mostly round, we call them coccus. And one way to remember this is it has the O in it, which is round, and so coccus is the round shape. If they're mostly kind of longer than they are wide, we would say that these guys are rod-shaped. These are also known as being bacillus. Another way to remember this, if you'll notice I have the two L's highlighted. This is because if you have the two L's, they look like little rods. You have two rods in there, so bacillus is the rod shape. The last one is the helical or spiral. We call this the spirillum. These are also known as spirochetes or vibrios. They kind of look like commas, but they are going to have more of like a curled kind of structure, kind of like a slinky or a spring. Okay, and that's the spirillum. Now, of course, if there's more than one, we call it cocci, bacilli, and spirilia instead of spirillum. So there is going to be the plural versions of those as well. Now let's talk about arrangements. And so if we're looking at the arrangement of these bacteria, they are going to arrange based on a lot of times what species they come from. And so if we're looking at cocci, these are all round, right? So these are the coccus. We're going to see that there are some special arrangements that are present. They can be in twos, and if they're in pairs, we call them diplo. So you would put diplo in front of the shape. So this would be diplococcus or cocci. On the other hand, strepto is where they're in a chain. Strepto means a chain, so this would be streptococci or streptococcus, where they're in a chain. They could be in tetrads. They could also be stacked tetrads. These guys are going to be when they come in fours, and they might shape kind of like cube-like structures. These are known as like packets, all right? They don't always get like the special names like we saw with the streptococci and the diplococci. Now, if they come in clusters, we call that staphylo. So staphylo is going to be where it kind of looks like a cluster of grapes, and so we call that staphylococcus. Now, the reason I hit on these is because there's not as many arrangements in the bacillus or spirochete or spirillum groups. They're kind of just on their own. They don't kind of merge together like these guys do and make certain arrangements. There are a few, but this is one reason we focused in on the coccus group. So organisms that are grown on artificial media, like we talked about last class, where we grow them on plates, and those plates then allow the bacteria to grow, they don't normally show the cellular arrangement that they would if you were to take the culture from, say, somebody's throat or a wound or a surface. They are going to not show that shape as well because they have all this space and they have unlimited resources, so they may not actually have that structured arrangement. So you may not see streptococcus actually in chains, okay? So here's an example. You don't see it necessarily in chains completely. Or when we look at staphylococcus, you may not see it in clusters like you normally would. And we do see, remember, this is gram staining from the agar plate, and so you can see that staining purpose, okay, so that you can see them better. So I'm going to have you describe the following by their gram reaction, which we learned in the previous video, the morphology, and the arrangement. So if we look at this first one, you'll notice we're going to be focusing on those little purple structures. Since they're purple, remember that is gram positive. If you'll notice, these guys are in pairs, so that's the diplo, and they are mostly circular, so this is cocci. So this is gram positive diplococci. Now, on the other hand, the next one is also in pairs, mostly round. They are a little longer where we might think that they might be bacillus, but they're still a little too small. This is going to be in pink though. The pink tells you that it's gram-negative, but it's still a diplococci, okay? Now what about the last one? The last one over here is in the purple, so that tells us it's gram-positive. They are cocci shaped and they're in chains. Okay, and so technically we could potentially say that these are streptococcus. Now, this is not the definitive ID of a bacteria. We cannot completely be like, yep, that's exactly what that bacteria is just by looking at its shape, which is the morphology, and its gram reaction. We would need to do further tests in order to know the exact type of bacteria that is present. And we're going to be talking about these tests that help us identify bacteria a little later on in the course. So when we look at this is why we can't necessarily say that this is Streptococcus pneumoniae. We might think this might be Neisseria gonorrhea. We're not really sure. This could also be Enterococcus. We would have to do more tests in order to find out. All right, so let's look at some of the arrangements that bacilli will do. Remember, they don't do as many. And so when we look here, we do see they a lot of times will come in that single bacillus form that you see in that first picture. And sometimes they will come in what we call the diplobacilli and the streptobacilli. But these terms are typically not used to describe the cellular arrangement for bacilli groups. So we don't necessarily call them diplobacilli or streptobacilli, like we saw with Diplococcus or Streptococcus. The term bacillus is used to describe the cellular morphology, the shape, which is not the same as when you look at the bacillus species when it is capitalized and italicized. Sometimes we use it as the species name of something, and that's capitalized and italicized, but if it's not, we are just talking about the shape of the bacteria. Another one you can see here is the coccobacillus. These are the ones that are kind of in the in between. It's hard to tell if they're coccus or if they're bacillus, and there are a few bacteria that fit into that group. All right, so back to describing the morphology. Can you describe the following by their gram reaction, their morphology, and their arrangement? So if we look at the first group here, they are mostly purple, and so because of that, it is gram positive. You'll notice they are rod-shaped, so these are bacilli, gram-positive bacilli. And sometimes they name these as looking like boxcars. Like if you were looking at a train from above, and they look like those boxcars of the train for transport. The next one, when we look at it, it is also purple, so it is gram-positive. They are bacillus, but they are in chains. Now, I mean, technically we could say that these are streptobacilli, but again, we don't normally use those terms. Now, if you'll notice though, these guys all have a very uniform type of morphology or shape, and so these are known as monomorphic. Mono meaning one, morphic meaning shape, okay? But if you look at this last picture, a lot of these actually kind of look like a club. They have like one end that's a lot wider than the other. These guys are described as looking like Chinese letters or clubs, okay? And they are gram-positive, but because they all don't have the exact kind of same structure, they're known as paleomorphic bacillus. Paleomorphic means there can be many shapes that go with them. An example of these are the diphtheroids, more specifically the Corneum bacterium. This group is going to have this kind of shape-shifting type of structure. So they're still kind of in the bacillus group, but they are not as uniform as we see with the other two examples. All right, so let's talk about the structures that are external to the cell wall that can be found in bacteria. All right, so we have plasma membrane, then we have the cell wall, and then these are outside of the cell wall. All right, and so we're going to kind of look at these. Now, the outermost layer is known as the glycocalyx. It's a capsule. This capsule is going to what gives bacteria some of its virulence, meaning that it can cause disease, and it's firmly attached to the cell wall. Now, if it's loosely attached to the cell wall, we call it a slime layer. But they are both this outer layer that's in the orange. So let's take a look at these three layers that are present. We have a yellow layer, a green layer, and an orange layer. So the innermost layer is that plasma membrane. So PM here is just our abbreviation for plasma membrane, okay? The green layer is the cell wall. So in a lot of cases, remember this has the peptidoglycan that is present. And then this orange layer is what we would call the outer capsule. Now remember, if it's more tightly attached, we call it glycocalyx or a capsule. If it's loosely attached, we call it a slime layer. Now, a lot of times this outer layer does create a gelatinous kind of sugar coat. This sugar coat protects the cell from being engulfed by like your immune system. Again, this is what makes it more virulent. Your immune system can't grab a hold of it to destroy it, and so it's going to allow it to stick around longer. Sometimes there's also going to be a K antigen. This is a marker on there. This marker is going to be a capsular antigen, and it's going to induce an immune response from your immune system. All right, because your immune system is going to notice that that does not belong, and it's going to then try to attack it. So what type of staining is used to see capsules? We talked about this in the previous video, and remember it shows that there is kind of like this highlighted area around the bacteria that doesn't get stained. This is known as a negative stain. All right, and so this one is known as a negative stain, sometimes known as is used with Indian ink. All right, so let's take a look here at some of these plates of organisms who have a kind of capsule or slime mold to or slime layer to them. If we look here, there's the Klebsiella pneumoniae. This is being grown on a type of plate called a McConkey auger. You can see that it looks very kind of sticky and mucousy as it's growing on that plate. Another one here is the Streptococcus pneumoniae. This is being grown on a 5% sheep blood auger. So the auger has sheep's blood in it. That's why it's red. And it also has this mucousy type structure to it. So the colony morphology gives what we call a mucoid look on the plate. And some virulent strands of Bacillus anthracis also produces capsules as well. Now remember, capsules allow these bacteria to stay dormant for long periods of time, and especially when we see that conditions are not favorable for them. All right, and so it's a form of protection for the bacteria as well as a way for it to try to evade your immune system. The next structure that's found outside of the cell wall in a bacteria is the flagella. Flagella is used for motility, for movement. And we do see that it is associated specifically with what we call an H antigen. So if you end up seeing some sort of marker on a bacteria where it has this H and then a number, a lot of times it's referring to that antigen. And this is what's going to elicit, again, a response from your immune system. So an example here is one of the E. coli substrands, or what we call a serovar, which means just like a subtype. You'll see it says O157:H7. That H7 is talking specifically about the type of flagella that's present on that E. coli. And a lot of times it's what makes it more virulent, and that's the E. coli that can make you sicker. All right, and so there's different arrangements that the flagella can be on a bacteria, and this picture shows you 4 different arrangements. The first one is the peritrichosis. The peritrichosis is going to be where there's many flagella and they're all over the bacteria. So there's a ton of them and they're all over the bacteria. On B, you see that there's the monotrichosis, and it's polar, meaning it's on one side of the cell, and there's only one of them. All right, so mono tells you there's one, polar means it's on one side. The lophotrichosis is going to be where it's on one side, okay, it's polar, it's on one side, but there's more than one flagella. So there's a lot of flagella, but they're all on one end. And then the last one is the amphitrichosis, and these are also polar, but they're on both sides. The flagella actually runs through the whole part of the cell. And I'm going to show you a closer picture of this in just a minute. All right, but the flagella is on both sides, but it's technically one long one connected through the whole cell. Okay. And this is what a lot of times gives those spirochetes a rotational or corkscrew type of movement as they move with this type of flagella. So taking a closer look, we do see that these axial filaments, the ones that run like on both sides, will a lot of times look like this as well inside of or around the bacteria. These guys are found mostly on the spirochetes, like with what causes syphilis. Treponema pallidum is the causative agent. It's the bacterial name. And these guys have what we call endoflagella. These guys run inside a sheath around the bacteria, and these wrap around the cell, and they will actually help them move in that corkscrew type of structure. This allows them to move very easily through fluids. Another structure we see is the fimbriae. The fimbriae is going to be shorter than what we saw with the flagella. They are going to be a lot of times thinner, okay, than the flagella as well. These guys are also going to be used for adhesion, like to hold on to surfaces, as well as they can create a structure called a sex pilus. The sex pilus is a fimbriae that gets longer, and it actually is more of a tube. When it attaches to another bacteria, it can transfer genetic information through that tube. So they can share pieces of their genetic information. They just can't give their whole DNA. And so a lot of times with the adhesion with this fimbriae is it gives it a kind of Velcro-like structure to it. They have these bristle-like fibers. They normally will have about 200 to 400 of them per cell, and again, it allows them to stick to surfaces. If these guys are present, a lot of times they're associated with gram-negative organisms like with E. coli and Neisseria gonorrhea. So these are some examples. So the next structure we want to look at is the actual cell wall. The cell wall protects the plasma membrane, and it gives the cell its particular shape, whether it's coccus, bacillus, or spirillum. We do see that if it stains gram positive, it has a thicker layer of peptidoglycan, which is kind of shown in the brown. This is going to allow it to hold on to the crystal violet stain, and the gram positives appear purple under the microscope. On the other hand, if they have gram negative layers, this is going to be where the peptidoglycan layer is a lot thinner, and we see that they have a lipopolysaccharide layer. This lipopolysaccharide layer is a fat layer, which causes them not to be able to hold on to the crystal violet as well, and so they appear pink due to the safranin stain. We do see a lot of times that LPS, this lipopolysaccharide, is also going to have a lipid A component, and what's crazy about this is that this lipid A, when the cell dies, say your immune system destroys it or it dies, it releases this lipid A as an endotoxin, and it can still cause you to have symptoms and still feel sick, even though you've taken care and killed off the bacteria. They also have an O polysaccharide. This is a type of antigen that is found on E. coli. So we talked about how the H7 was the flagella. The O157 is talking about this particular part of their cell wall. Okay, and so again, this is what helps make this bacteria of E. coli more harmful to you, is the cell wall component of O157 and the flagella component of H7. Now again, remember there are some exceptions to the rule when we talk about the peptidoglycan. These are going to be the mycoplasm type of cell walls. It is the causative agent of things like walking pneumonia, and they lack a cell wall. They don't even have one. These guys are the smallest living microbe because they're not a virus. It's an actual bacteria, and they have a very high lipid content in their plasma membrane. Mycobacterium also have a high lipid content, and we call that myclonic acid in their cell wall, and they cannot be used with gram staining. We have to use those acid-fast staining, that AFB staining, in order to see their cell wall. Archaea cell walls are different than bacteria, so again, that's one reason why they're in a different group, and we don't really talk about them here. And the cell wall is one of the key areas that some antibiotics will target. So some antibiotics will actually interfere with how cell walls are made, and this is how they can breach and get through that bacteria and destroy it. An example is penicillin. Penicillin is effective against a lot of gram-positive bacteria because of the peptidoglycan layer, and it's able to help poke holes in that, which allows your immune system to fight it better. So some concepts here before we get into some gram reaction examples. Bacterial cells with atypical cell walls, the ones that are different types of cell walls, they don't gram stain very easily. And so because of that, we have to use different staining techniques. Different antibiotics are used on gram-positive versus gram-negative, and this is due to the structural differences of the cell wall. So not all antibiotics are going to work great against every type of bacteria. And damage to the cell wall can lead to what we call osmotic rupture of the plasma membrane, where the cell actually can be destroyed, right? And so poking holes in the cell wall allows your body to help destroy that bacteria. All right, so let's do a little comparison here of gram-positive versus gram-negative. Remember with the gram reaction, the gram-positive appears more purple because of the crystal violet, whereas the gram-negative is going to absorb the counterstain, which is the safranin, which gives it a pinkish red color. Peptidoglycan layers in gram-positives, they are thick. In gram-negatives, they are thin. We do see outer lipopolysaccharides or endotoxins being released. In gram-positive, that's absent, but in gram-negative, it is present. We'd see sensitivity to lysozymes in penicillin is high with gram-positives, but low with gram-negatives. So penicillin or penicillin derivative antibiotics are not good against gram-negative bacteria. A resistance to drying out is high with the gram-positives, but it's low with the gram-negative. The gram-negative cannot be on a surface or remain on a surface for as long. So let's now switch gears a little bit and let's talk about the cell wall and some of the inner structures of the prokaryotic cells. So when we look here at the plasma membrane, this plasma membrane is also known as the cell membrane or the cytoplasmic membrane. Those terms are kind of interchangeable. The structure of the plasma membrane is known as the fluid mosaic model, and this is because it's made out of these structures that you see in the orange and yellow called phospholipids. So the PL there stands for phospholipid. These phospholipids are going to have a polar end and a non-polar end. So the heads in the orange like water, and the yellow tails are hydrophobic and they don't like water. All right, and so this is what creates this bilayer, so two layers of these fats that form the plasma membrane. Inside of the plasma membrane, you do find there's a lot of these proteins. They're shown in kind of this blue color. Some of these proteins are integral proteins, meaning they run through the whole membrane. Others are peripheral proteins where they're just on one side or the other, kind of like your peripheral vision. Now, bacterial plasma membranes do not have the following. They do not have the glycolipids, the glycoproteins, or the sterols. Now, there is an exception with the mycoplasms. They do have the sterols that are present, but they are not going to have most of these extra structures that you find on eukaryotic plasma membranes. So what's the function of the plasma membrane? Well, the function is that it acts as a barrier between the inside and the outside of the cell. So it separates the internal environment from the external environment. We also see that it is subject to rupture, so it's not actually protective like the cell wall is protective. If too much fluid gets inside of the plasma membrane, inside the cell, and it expands, the plasma membrane will rupture or bust, kind of like putting too much water in a water balloon. The cell wall helps prevent this. Transport substances into and out of the cell can occur in the plasma membrane, so we can actually send organisms out, bring things in. This could be chemicals, this could be water, this could be just anything that the cell may need. Plasma membrane is also selectively permeable, so it picks and chooses what gets to move in and out. It doesn't just let everything. So it's going to be very picky about what moves into the cell and what moves out of the cell. Now, the bacteria plasma membrane is also where cellular respiration is going to occur. So CR here is cellular respiration. This is how the cell gets energy because they do not contain the mitochondria, which is the organelle that creates ATP. They don't have it. So the cell membrane has proteins present that help them do this. Now, how do they transport things across the membrane? Well, sometimes they have to do active transport. If they do active transport, this is where energy is required. The form of energy that your cells use or bacteria cells use is ATP. This means that it creates more of like a pump. So you're going to use energy to move something in or out of the cell. Passive transport, on the other hand, does not require ATP. And so this is going to be where molecules move from a high concentration to a low concentration. And these molecules will do the movement without the plasma membrane having to exert any energy. So if we take a look at this picture here, you can see simple diffusion, which is a type of passive transport. These little orange molecules are moving from the high concentration outside the cell, and they're going to move into the cell to a lower concentration. We also see these next two pictures are also a type of diffusion, which is passive transport. This is facilitated diffusion though, and they are going to use these proteins that go through the membrane for help. So there's no ATP used, but they have to have a bridge or a transport area to help them get across. And so you'll see there's channel proteins and then there's carrier proteins. And they're going to help move those substances from high concentration to low. All right? The last one over here that is a part of passive transport is osmosis. Osmosis is the movement of water. And this is the movement of water from high to low concentration. Now, osmosis is this movement of water from high to low across the membrane. Well, what determines if it's high or low? That's the tonicity. The tonicity of a solution describes the solute concentration, what's dissolved in it. So if there's a lot of stuff dissolved in the water, we say that it is a hypertonic solution. So if you notice in this picture, there's a lot of those little green structures. There's a lot of them outside of the cell. It's hypertonic, okay? More solute, less water. On the other hand, if there are less solutes out there in that water, it's called hypotonic. Hypotonic is less solutes and more water. And the last one is isotonic, where everything is equal. It's the same amount, okay? And so we see that they have equal amounts of solute inside and outside of the cell. So why are we talking about this? Well, osmosis is determined by the solution that the cell is in. So in the different situations, water is always going to move from po to per. So it's always going to move from hypotonic to hypertonic. So in this first example, water is going to move out of the cell. See how it's moving from po inside the cell to per outside of the cell. This is going to cause the cell to shrivel up because it's losing water. On the other hand, in the second picture, water is going to move in because it's moving again from po outside of the cell to per inside of the cell. This is going to cause the cell to swell, to get bigger. In the isotonic, though, it's going to be equal. Water is going to move in and out at the same rate. If 2 move in, 2 move out. 5 move in, 5 move out. And this is because it is in equilibrium. So cells placed in an isotonic solution have no net movement of water. So again, 5 move in, 5 move out. There's no change in that. But cells placed in a hypotonic solution will swell. This is an osmotic lysis that can occur if the cell wall is damaged. And this is why antibiotics that damage the cell wall are important, because it can cause us to destroy the cell in this way. Okay, so antibiotics can help with this, as well as lysozyme activity, which is an enzyme that some of our cells have. On the other hand, if cells are placed in a hypertonic solution, they will shrink. This is known as plasmolysis. And so it pulls the plasma membrane away from the cell wall, which is also not good for the cell. So what's the application of this? Like, why is it important for us to know if water moves in versus water moves out? Well, the application part of this is controlling the growth of the microorganisms. Can we control whether bacteria grows there or not? This is going to be one of the things we'll look at in a future chapter as well. So they need perfect osmotic conditions for a bacteria to grow on the media. So if we want to grow it in the lab, we have to obviously make sure that it's growing well and has the right conditions. Bacterial cell walls in a hypotonic or hypotonic solution are subject to becoming metabolically inactive, which can create a problem. All right, and so this can control the amount of growth. So some other structures that are found out inside the cell are the cytoplasm. The cytoplasm is composed mostly of water. This does have a variety of inorganic and organic substances, which we'll learn more about in a following chapter as well, what that means. But these are chemicals. We also see that it is made out of an aqueous solution, so it has a lot of water that bathes the DNA area called the nucleoid and the ribosomes, which look like those little sprinkles inside of the cell. Now the nucleoid, remember, is the area that contains the single loop of double-stranded DNA, and that's called the chromosome. So the nucleoid area is going to be containing the chromosome. Outside of the chromosome, there may be these areas of DNA called plasmids. Plasmids are accessory rings of DNA. These are not critical for the metabolism of the bacteria, but they are extra genes. These extra genes can be donated to other bacteria through the sex pilus we talked about. They can actually be responsible for what causes antibiotic resistance in some bacteria, like in MRSA. Some of these plasmids also code for things like toxins that the cell can release, again, making us sicker from it. And we do use these plasmids a lot of times in biotechnology. We use them for gene manipulation. When we want a cell to take in that extra DNA, we use a plasmid to do it. So one example of what we do with this is we have a plasmid that has the information that codes for human insulin. Okay, it's the code for how to make insulin. They put it into bacteria and have bacteria make insulin. They then can package it and they can sell it at the pharmacy. All right, and so this is where the bacteria is making human insulin, but it doesn't need it. But we got it to make it by inserting a plasmid with that genetic information on it. All right, and that's what we see with biotechnology. Another structure that we see here are the ribosomes. Ribosomes are responsible for making proteins for the cell. So they are going to be responsible for what we call protein synthesis. The S that we see in that 70S stands for the Sevberg units. This is a measurement of their sedimentation rates, and they have a sedimentation rate of 70S, and so that's why they are a little different, because in eukaryotes it takes a little longer and it's 80S before they actually have their sedimentation rate. These guys have two subunits to them. They are named the small and the large. Okay, some antibiotics will actually target these ribosomes and cause them not to be able to do their job and make proteins. Again, this puts the cell at risk, and your immune system can then help destroy it. Okay, prokaryotic ribosomes are smaller than eukaryotic ribosomes. This means that antibiotics that can attack the smaller ones actually leave our ribosomes, the larger ones, alone. That is a good thing when we are picking medication. We want medication that's going to destroy the invader but not hurt us. And so a lot of these medications are good like this because they attack the 70S and not the 80S. Another thing we want to look at is something called the endospores. Endospores are metabolically inactive structures that allow cells to become dormant. Okay, so this is where conditions are not good, and so they can encapsulate themselves in these endospores, and they can remain dormant for a long time. These are what we would call dehydrated cells. They have thickened layers of peptidoglycan, and the purpose is for them to survive, not to reproduce, just to survive. This can be where they survive for years and years and years. They're very hardy, They can survive in the environment, but they're not going to actually grow and divide. They're just surviving. Once conditions are good again, they can actually start germination again, which means the cells can return to their vegetative state and they can actively start to do their metabolism and actively reproduce again. All right, so this is a kind of defense mechanism that some bacteria have when conditions are not good. So concept here, we must be able to kill the endospores during sterilization processes. If we don't, and then we put that instrument we sterilized into a person, those endospores could be transferred and then those conditions could be good and they could start growing again. So the sterilization process has got to get rid of endospores as well as the vegetative bacteria. So some examples of organisms that make endospores. We do see that some gram-positive endospores, endospore producing bacilli are listed here. Clostridium is one of them. This is going to cause botulism, tetanus, and gas gangrene, all which are terrible for us. We also see there's some bacillus species, and some of those bacillus species are what causes food poisoning as well as anthrax. So these are some examples. We see that these endospores can be on the edge, which are the terminal endospores, which you see here that makes them almost look like a lollipop. Whereas others can be in the middle of the cell, and those are called the central endospores. So some implications here when we're talking about these endospores is you have to make sure if you're preserving food like canning, you'd use the process correctly. If you don't follow the process correctly, then you may introduce endospores into the canned goods, and it can actually then make you sick. We also see that these are going to be important for vaccines as well as making people publicly aware that there's an issue if there is. So say we see that there's a recall on certain canned food goods that were not canned properly because they contain endospores. We need to work on getting those off the shelves, getting people to throw them away if need be. So public awareness is very important with these. All right, last thing here is the eukaryotes. We are going to focus on these when we get to the next video. But these guys are going to be the protista, the fungi, and the animalia. One thing to note is some structures are the same that these guys have with prokaryotic cells. They do contain flagella, cilia, and cell walls at times, but their structures are quite a bit different. Another thing that makes them more complex, remember, is they contain membrane-bound nucleus and organelles. And so you need to make sure that you review those organelles. You should have learned the organelles of eukaryotic cells back in a biology class. So you want to make sure that you review those because I'm only going to hit on a few of them in the next video. If you have any questions, please let me know.